We're going to look at mitosis. Now, this is a process of cell division, um, or of cloning of cells rather than of reproduction in terms of sexual reproduction. So, mitosis occurs in asexual reproduction. So, cloning, such as plants, how when you take root cuttings, etc., that would be cell division because it's a replica of the parent plant. So there are a few main distinctive steps in mitosis, and we'll go through them all one by one now. So we're going to start off at the first stage, which is known as interphase. Yeah, this is interphase, and in interphase, you've got your cell with all the organelles inside. And you've got your nucleus, and all the DNA is all randomly distributed around inside the cell. Okay, so this is basically the cell's neutral phase. That's how it is when it's not replicating through mitosis. Okay, and then and then the next phase is called prophase. Okay, and in prophase, what happens is you've got your cell, and two main things start to happen. You've still got all your organelles, and they're not doing much at the moment at all. And then you've got your nucleus, and two things are happening here. The first thing that's happening is the nucleus wall is becoming less distinct from the rest of the cell, so it's kind of dissolving and breaking down to some extent. And the second thing that's happening is the random DNA, which is just sort of randomly spilled around in the molecule, in the nucleus in interphase, is now actually collecting itself into actual chromosomes. So you've got the central centromere around which you've got your chromatids, and these are actually your chromosomes because the DNA is coagulating into your chromosomes. And they were always sort of in this structure, but they were so loose that it was almost impossible to distinguish the chromosomes. In fact, in images of cells, it would be impossible to distinguish the chromosomes until they come to prophase, in which case they will become distinct chromosomes. Okay, and then the two things that happen in prophase. Now, in metaphase, we've got at this point, the cell wall, the nucleus membrane has completely dissolved. It's gone, so we've got no distinctive nucleus in the cell. We've still got the outer cell wall intact, and it's an original structure. We've still got our chromosomes inside. Just to simplify things, we'll just take an example with a cell with 2 chromosomes. Although that's very simplistic cell, it's easy enough just to scale up with more chromosomes, but it's just easier for me to draw and to explain with the 2 chromosomes. Okay, and what happens is spindle microtubes extend out from either side of the cell, and they go through the centromere of each of these chromosomes, and then they attach each end of the cell. And there's lots of these spindle micro tubes, but not all of them attached to the chromosomes. Okay, and then that's metaphase. Now, if we go on to anaphase, we've got our cell again, and we've still got this time at the centromere, the chromosome splits apart into its chromatids. So we've only got half of the chromosome here because they've split apart as the spindle microtubes draw them to either side of the cell. And we've still got the spindle tubes aren't attached to the. There you go. So they started to pull the chromatids apart so they come to either side of the cell. So each half of the cell has got exactly half the genetic material, but the genetic material is identical because bear in mind the genetic material in this chromatid that is split apart is the same as that. So they are identical. So each half of the cell is getting half the genetic material, but the genetic material is identical because there are copies in the middle. So now we've gone over anaphase. Now if we move on to telophase. Okay, and at this point the cell is starting to split into two. You can see its shape is changing, and then we've got our chromatid at either end, and all the spindle microtubes have now all vanished. They've all dissolved into the mitochondrial matrix. Okay, now because the total surface area of the two new cells is going to be more than the original membrane that was around the mother cell, what's going to happen is more membrane is going to have to form, and that membrane is normally formed from the endoplasmic reticulum. Okay, so that starts to form a membrane across here. Now, the final phase in mitosis is psychokinesis, which is basically where the cells are almost completely divided now, and you've got your nucleus and all the chromosomes have gone back to being in their normal, really spread out, visual recognizable state. But at this point, all the organelles in the cells in the mitochondrial matrix now split apart. Now that all the organelles, there's multiples of them, but they don't exactly split apart equally. So what basically happens is they split apart randomly, and you'll get approximately equal amounts of each organelle in each cell. It doesn't have to be exact, but as long as there is at least one of each organelle in each cell, and occasionally this goes wrong, but most of the time it doesn't, but because it is random, so there is chance for error here. But normally it doesn't, and they're just divided randomly between the cells. And of course, at this point, we are back at interphase again, which if we go all the way back, so here we go, we're back at interphase where the cell is in its neutral state before it starts to split. But this time we've got two. If you found this tutorial useful, don't forget to hit the like button below. If you've got any questions, post them in the comments box. And thanks for watching.